So our next speaker is Dr. Jeff Chen. He's joining us virtually. Dr. Chen's research is helping us better understand the cellular mechanisms involved in LCC, including how the integrated stress response may contribute to the disease today. He will share what his team is learning and how these findings could help guide future therapeutic approaches. Thank you for joining us.
Hello, everyone. I would like to thank Ashley for her introduction and organizing this wonderful symposium. My name is Jeff Chen. Today, I'm going to talk with you about the model and the mechanism of LCC caused by SNORD118 mutation. I'm a professor and associate dean of research at Austral School of Dentistry, University of Southern California. I serve as the director of the craniofacial PhD program, as well as associate director of Center for Craniofacial Molecular Biology. So my lab is study the interface between brain and skull, and but particularly how the skull and the brain interact with each other, how dysregulation can lead to the craniofacial neurological disorder, such as microcephaly, craniosynostosis. In addition to this basic research program, I'm also leading a national clinic translational research program called USA TMD Impact. So we aim to improve the patient-centered translational research, focusing on this temporomandibular disorder, the joint, which is very critical for our smiling, speech, and chewing every day. And this Better TMD program is one of the 3 research teams funded by NIH and DCR to support clinical translational study, which I think probably it's something can employ this model for future clinical translational study. So in this particular study, our USC site, we recruit about 2,500 patients, and we have patient sign symptom, we have clinician structure note, we have patient self-report treatment outcome. And meanwhile, we will employ a multimodal, the deep phenotyping, including the saliva, the synovial fluid, as well as the DNA genetic analysis, such as DNA methylation, such as the single nucleotide polymorphism study, as well as the iPSC-derived organoid of TMJ organ-on-chip model. So we wanted to integrate all those patient data with the lab data and use machine learning, deep learning, AI-powered tool to build the precision medicine platform to inform the diagnosis and treatment for clinician for this disease. Again, I feel this could be used in the LCC study. And particularly, I like to emphasize that those type of study is very much partner with our patient. In this case, the patient is not just like a study subject. More importantly, they actively, directly involved or direct our research. So how we do that? So our patient engagement, first we have partnered with TMJ Association and as our advocacy group. And meanwhile, we formed the patient advisory board with the people with lived experience, disease experience, who we are joined our monthly meeting to lead and guide our research and provide feedback about our research, like patient burden, and how the patient feel satisfied by the clinical study, and also help us to disseminate the data back to the community. Back to the SNORD118 study, we know that the ribosome is very important to translate the mRNA into the protein. As we all know that while our nerve can fire, while our heart can beat and the muscle can have stress, it's mainly because the protein are doing the job. Those protein need relying on this ribosome machine to translate the mRNA information, which mRNA is from DNA, and mRNA information need to be translated into protein. That's what the ribosome is doing, translation from mRNA to the protein. SNORD118 includes this non-coding RNA, box CD non-coding RNA. And as you can see here, it's a very complex structure and they can self-fold. And this non-coding RNA encoded by SNORD118 is very critical for this ribosome biogenesis function. And without it or impair this SNORD118, which is the genetic cause of the LCC disease, can impair the ribosome biogenesis function, leads to the protein translation defect, and that's why there's disease occur. If we look at the literature on and published the patient case, a variety of different mutation occur in the SNORD118 gene. And one of the shared symptoms for the disease is this hyperintensity of the, also we call the white matter defect, and as well as the other like vascular defect we call microbleed, and also increased blood-brain barrier permeability calcification, in addition to this immune dysregulation inflammation, which probably involves microglia, astrocyte function. However, we don't know what kind of cell type are particularly sensitive to this SNORD118 mutation caused the ribosome biogenesis. This is by itself, it's an important scientific question, is because ribosome biogenesis is required universally for all the cell to make the protein, right? But why some cell, some are particularly sensitive to the mutation of SNORD118? To answer that question, our lab started to make these SNORD118 mutant mice, first of all, to see whether they can recapitulate some morphologic phenotype in the patient, which indeed is the case. In some patients, we found they have a relatively brain smaller size. And more important, we wanted to find out, use those in vivo animal model to find out what kind of cell type are particularly sensitive to the mutation, to the disruption of ribosome biogenesis. Are they neuron, or are they immune cells such as macrophage, microglia, or they are like blood vessel cell, which are still ongoing in the lab. So I want to share with you that indeed we found that there's this white matter hyperintensity in our mutant mice, as revealed by this MRI analysis, which was done by our student research assistant Alexia, who is on this Zoom meeting. And if we look at the different regions such as white matter, we can clearly see the mutant mice. We can see increased hyperintensity in mutant mice. Meanwhile, Alexia study through the MRI started to show that there's increased blood-brain barrier permeability in mutant mice compared to control mice. And again, this suggests that there's a broad vasculature issue. We actively pursue those study, those direction. So those in vivo mouse model can allow us to study the mechanism in vivo function. We wanted to step further to trying to understand mechanism, particularly linked with human mutation, and how that can be used as a platform to do the drug screen. For that purpose, we turned our attention to this induced pluripotent stem cell iPSC system. As we all know that the patient cells such as fibroblasts or blood cell, mononuclear monocell, can be induced through the Yamanaka factor to this pluripotent stem cell. So those cells can differentiate any cell type in our body. Could it be cardiomyocyte, could it be neuron, could it be immune cell, could it be any other type of cell. And as I mentioned before, through the animal study, we can identify which cell, such as neuron, could be particularly sensitive to this SNORD118 mutation. Therefore, we can use this human iPSC platform to differentiate this progenitor cell into the cell type that are particularly sensitive to the SNORD118 mutation. And also, people started to have developed this organoid model, like brain organoid model. It's not just differentiate into the neuron. They also can model grow into structured with proper organization of cell type within this organoid, very much mimic the human brain development and structure. And use that, we can screen the drug, the FDA approved drug, basically repurpose those drugs which have been used in other disease and never be used in the SNORD118. We can start to repurpose those drug, potentially rescue the phenotype in the iPSC cell model, organoid model, as well as the in vivo animal model before the clinical trial into the human. Our lab has been extensively studied this brain organoid to model the different diseases such as microcephaly, such as autistic phenotype, and we have extensive experience to generate the human iPSC and culture then started from single cell grow into the brain organoid. So that's what we did for the SNORD118 project. So we use CRISPR-Cas9 genome editing approach, introduce this particularly patient mutation into SNORD118 human iPSC, and from compare the control and patient-specific mutant iPSC derived organoid, we found there's brain size difference, and we analyzed their phenotype at a cellular level. We found there's decreased progenitor cell in the mutant organoid, decreased cell proliferation. That can explain why the brain size is different in the mutant. And more importantly, we can use those cellular phenotype as an assay, functional assay, as a screen platform to screen those small molecule compound to find potential the drug to treat this kind of defect. So in this particular case, we identified this integrated stress response drug, small molecular, to be act as a potential target to treat this disease, starting from the rescue the mutant phenotype, the brain organoid size. And overall, I feel there's a lot of opportunity to integrate the research with the patient data, patient care. For example, we can start from the patient blood collection and use the mononuclear cell from blood to reprogram into the human iPSC. And from human iPSC, we can grow then into brain organoid or other cell type particularly sensitive to the SNORD118 nuclei, such as a blood vessel cell. And from there, we can diagnose or predict disease severity and progression by analyze those brain organoid or iPSC-derived macroglia or like vasculature cell. And second, we can study the disease mechanism to find out why SNORD118 mutation can lead to different disruption of different cell type, cause the disease, identify downstream the signaling pathway, molecular mechanism, cellular mechanism for the disease. And last but not least is that we can use those phenotype as a platform also use this iPSC-derived cell type of organoid as a platform to screen the FDA-approved drug for potential treatment. And those drugs can be used in the organoid in vitro model and then come to the in vivo model for the behavior assay, the brain morphology, as well as the neurophysiological assay, and ultimately can help transition for the potential clinical trial study. So I will end here. Feel free if you have any question about the study.